Hello and welcome to this teaching ideas video on how to use a large cell cycle model at GCSE or at A-level biology. Right, the first thing to do is to go and build a large circle that you can go and place in the middle of the table. Right, you might want to go and put arrows on this depending on how you want to do it. Section off different parts of this circle in different colours to represent the different stages during the cell cycle. After you've done that, you can go and make lots of little pieces that represent different things that are happening. So at each stage, you might want to go and produce different cells that show the state of the chromosomes at each particular stage. There are also some other key points like the chromosomal checkpoints and the G0 checkpoints, etc., that you might want to go and put little labels on to go and place on. Finally, make sure that you go and put lots of annotation cards on there that students can then go and use to go and produce their own cell cycle diagram from the model that you've used. And as always, as you're going through it, make sure that you keep pausing, questioning your students about why certain things are happening at certain times and what they think are the importance of it. The next short video will hopefully show how this model is used in practice.
Okay, yeah.
So we start off your cell cycle. You've got your cell, 46 chromosomes, which is 23 pairs of chromosomes. It enters the G1 phase. Right, G1 phase, you can either call it, technically it's called the gap phase, but if you want to think of it as your growth 1 phase. So this is where all of like your large structures are copied. So your organelles are duplicated. It's where you go and you're making sure that you've got enough phospholipids produced. It's where all those other little building blocks that are required are produced. So at the end of G1 phase, you've not duplicated any of your chromosomes, most importantly. So you still have the same number of chromosomes. So after your G1 phase, you move into the S phase. So the S phase, right, is called the synthesis phase. Right? During this point, this is where your DNA is copied. So you're going to go technically from 46 chromosomes to 92 chromosomes, so 46 pairs of chromosomes. Right? So we go and we copy the DNA in the S phase. After the S phase, you have the second gap phase. In your second gap phase, that's just where the cell is going to carry on enlarging. It carries on checking that it's got enough organelles. It carries on checking that it's got enough other biological molecules. So if we go and we put the G1 phase, the S phase, and the G2 phase together, all of those three are classified as the interphase. Right, all interphase means is in between mitosis. Right, it's probably the easiest way to think about it. So at the end of the G2 phase, we move into your M phase. Right, your M phase, or your mitotic phase, is where your mitosis is going to happen. So it's where the DNA is going to get pulled apart and you're going to get cell division happening during cytokinesis. So all cytokinesis is the one cell splitting into two cells, right, which are both going to have an equal number of chromosomes. Right, you can almost think of it as cyto, a bit like cytoplasm, and kinesis to chop. The cell at this point can either go into a G0 phase, right, where it's not going to do much of anything, right, it'll just carry on, and it goes into a quiescent state, right. All quiescent means is it's not dividing, right. After a certain period of time, it can go from that G0 phase back into G1 and it'll carry on dividing. So that is your basic cell cycles. So the second features of the cell cycle that we need to consider are the checkpoints. So at the end of your G1 phase, there is a G1 checkpoint. At the end of the G2 phase, there is a G2 checkpoint, and in mitosis, there's a spindle assembly checkpoint. The G1 checkpoint goes and it checks the cell size to make sure it's big enough to divide, checks that it's got enough nutrients, it checks that there's enough growth factors, right? So we need the growth factors there to get it to carry on and grow after we go through cytokinesis, and it checks for DNA damage, because you don't want to go and start replicating DNA that's already damaged, or else you'll get mutations happening. Right, your G2 checkpoint, right, it goes and it carries on, it checks that it's got enough growth factors and nutrients and such, but more importantly, it checks that the DNA has not been damaged during DNA replication. Because during DNA replication, all of your DNA is unravelled from those histone complexes and it's spun out. So you could quite easily get a substitution happening, or a deletion, or something wrong with the DNA, right? If it turns out that your DNA is damaged, then either you'll go back into a G0 phase, or your cell might undergo apoptosis and die, right? Because you don't want it carrying on if the DNA is damaged, or else you'll have mutations. Last part, back end of mitosis, you've got the spindle assembly checkpoint, because if your spindle fibres haven't gone in attached to these pairs of chromosomes correctly, you might get incorrect number of chromosomes going to both sides, which can cause an entire host of problems and inherited diseases and such forth. So at that point, all we do, we just check that the spindle fibres are attached incorrectly. Right, stop.
Hopefully this video has given you some ideas how to teach this particular concept. If you've liked this video, please subscribe, like the video. There's links below to follow me on Facebook, Twitter, and there's a link below to my shop on test where you can go and get a full copy of this lesson. Please feel free to leave any comments or questions below, and I will get back to you as quickly as possible. Thanks for watching and happy teaching.